So the obesity is not only itself is a problem, it also is a cause for a lot of other secondary disease. I list here. Typically type 2 diabetes, that's almost always associated with obesity. And cancer, liver disease, and also some hypertension, mood disorders. So the obesity can cause a lot of secondary disease that adds up, can cause a lot of cost, not only talking about quality of human life, cost, this is a huge burden to our society. And the reason is that the food now become so delicious. If you get used to food, you like it, you cannot control. So how to control your behavior towards different food? That's a huge issue for some of us. So that today is a topic of how the brain control our feeding behavior. So this is on the obesity side, but on the other side is also a problem because the other spectrum of these people, we call it in the human condition, we call it anorexia. So they refuse to eat no matter how malnutrition they have. They suffer from extreme starvation. And then this disease has the highest rate of mortality because it's a mental disease. They control their behavior. Nobody else can control it. They just refuse to eat. They starve to death. Look at this image. This is an actual patient, very, very in a starved situation, very disturbing. But the reason is not because they not feel hungry. They feel hungry. But they still refuse to eat because mentally they think they are not under starvation. They always think in the mirror, although they are very thin, but they always think they are so fat. They worry about, they are so fat, always worry about. That kind of thinking dominate their behavior so that they always, once they start to eating, they always have that thought in their mind, so that prevent them from eating. So it's kind of a vicious cycle. So this is called a persistent uncontrolled behavior. So I will talk about in a animal behavior also show similar thing like that. So associated with that, they have a very negative feeling. So you talk about those patients, they're not happy. They're always worried about something. So this is a very dire situation for those patients, and because there's no cure for that. So now I just want to come back to the overall situation here. So whether there's no eating or eating, all we can say is abnormal eating. So you one situation, you have a behavior that you eat when it's not necessary because you have enough nutrient in your body, but you still want to eat. So that's a unwanted behavior. And always they associate with a hedonic feeling. They feel like food is so delicious, they cannot control. Once they see the restaurant and they go in, no matter what, no matter they just had food, they always want to get more food to eat. They have more motivation to eat because they try to come up with the excuse to eat those food. On the other hand, no eating when it's absolutely necessary. They refuse to eat. These normally come with a psychological disorders, anxiety, possessive disease, a compulsive disorder, because they have this compulsive thinking that they are fat. Actually, they are not at all. And they're not listening to the parents. The anorexia normally occur in teenage girls. They're always kind of fixated on their image and without effective treatment. So for us, we want to identify the root of the problem, where in the brain that goes wrong with this different behavior. So let's look at the feeding behavior. Feeding normally for most of us here, we feel like we feed, we feel hunger. And once we feel satiety, we'll stop eating. So this is called a homeostatic. So the brain is a homeostasis, always your body weight is reach a steady level. It's okay? Okay. But the feeding behavior is not only that simple. So the feeding behavior also heavily influenced by a lot of your higher brain function, your fear response, your stress. So sometimes you feel stressed, you lose appetite. No matter how hungry you are, you don't want to eat. Emotion, pleasure. I mean, the delicious food always give you a pleasure feeling, right? So this is all influenced by these different emotional states. And so here I give you an example about how to control feeding, right? So in this situation, this is about the animal. They go out to search for food. They prey other animals, but they're also afraid that they become victim of other predators, right? So there's always a balance to decide whether they go out to get food or not. Similar as this little guy, this one mouse, she faced that situation. If that little mouse, they're very hungry, how they decide to approach food is like eat or not eat is a survival issue, right? If you eat, you will get caught, you're dead. So this is a lot of environmental situation that can control our feeding behavior. Even now, as a human, we are heavily influenced by the environmental setting. There's a study that for human, if you eat same food outside the door, inside the door, those people eat a lot more food than people eat outside the door. Because outside the door, you have these consciousness, you always look out to see whether there's a danger coming towards you. So there's the humans still have that kind of innate behavior. It's, if you think about it, it's kind of interesting. So, so we believe that in our brain, because all these behavior, all this association, we believe that the feeding and those emotional behaviors are controlled by the same set of neurons or same neural pathways in the brain. So we wanted to study that. Here, I also want to give you, just to reiterate the scenario, right? You have this delicious food for this person. If he, in the brain, he always think, oh, I'm fat. That feeling gives a dangerous signal so that they will eat less. And if always think about a pleasure associated with food, they will eat more. And extreme conditions that if you have compulsive thinking in this dangerous signal, then you will have anorexia. And if this pleasure thinking dominated your behavior, then you'll have this compulsive feeling, and you'll have overeating, and probably you'll become obese. So this is the basic situation. So let's look at evolution, right? During evolution, our ancestors, thousands of years ago, and they spend most of their time hunting for food. There's no time for them to do other things because they just barely get enough food for them to get stomach filled. But nowadays, the situation completely changed. You can drive, go to a fast restaurant. Within a few seconds, you have this food that's more delicious. And that's the outcome. And that's the outcome for some of us, right? They be very obese. They get a vicious cycle. Once they get obese, you eat more food and you get more delicious as a vicious cycle. And that this is a mouse in the wild. They have to go out during night because during day it's too dangerous for them. So during night, they go out, they search for food, they spend a lot all day, all night long to search for barely enough food for them to survive. But now if you house this animal in the animal facility, in our housing room, the food is available 24 hours. And if they have different food, they'll become very obese. So from evolution point of view, the phenomena is shared. Definitely they have the same mechanism that facing this environmental change, they develop obesity. So we use animal model to study feeding behavior and related physiology. That's the rationale behind why we can use animal to understand our human brain and human physiology. So this just to give you a general background for a healthy individual, your food intake and energy expenditure remain equal so that your body weight remains stable. If the intake is more than expenditure, you'll become obesity. I mean, most of a young child, during development, they have to eat much more food than anything to allow them to grow. But for adults, mostly they are stable. If the intake is less than expenditure to a certain degree, you'll develop anorexia. And how we control that? And this is all controlled by the brain. So the brain receive a lot of feedback signals. And these feedback signals like hormones, nutrients from the blood that can reach to the brain. The brain neurons can sense it. And then the brain neuron can control our behavior. Two behaviors. One is feeding, how to control your feed. The other one is control energy expenditure, which is how to control your exercise, control your muscle, your temperature, that could control a lot of energy that you need to maintain your body temperature. So this is the brain control for that. So the brain is the basis for all the behavior, including eating. Let's look at our brain. Our brain is so complicated. We have 100 billion neurons. And not only the number is so big, and which each neuron, they have a lot of connection between them. And so this is a very complicated system. And each neuron, how the neuron work in our brain. There's two basic mechanisms. The neuron can be excited or can be inhibited. Just like our computer, right? Our computer 1010, I don't know, but the brain also have this kind of similar code. They either be excited, excitation, then allow your behavior to happen, or inhibited, that will stop your behavior. So something just like you can understand that way. So either the neuron activated or inhibited. It's just like a green light or red light, something like that. They use this basic modality in the brain. So our research is trying to identify key neurons in the brain that can control feeding behavior. So how to do that? Because brain is so complicated. So first we want to decide which region we want to target, right? So the region I will target is a region called hypothalamus. I don't know, you guys don't need to remember this, but we have a specific region called paraventricular hypothalamus. It's a difficult term, but we just call it PVH. We can just use PVH, simple. And this brain area function as a portal system to control brain and the peripheral organs, like all the peripheral organs. And previous study, people damage this area, they will get very obese. So that's the information about this one. So we want to study this as an entry point to understand the brain. So to understand the neuron, as I said, this brain, as I said, the neural activity is important for the behavior. So we want to control the neuron activity like at our will. We want to inhibit it, we want to activate it. So it allow us to see the consequence. So one way to do that is called optogenetics. You guys never heard about it, but it's simple. It's just like light plus genetic manipulation. So we can use this typical bioengineering approach. We can express some protein that can be controlled by light. We already know that identify in other species, like in this case in algae, in the plant, algae. Now we know that protein can be controlled by light, and we can express that protein using bioengineering method to specifically in these neurons. Then we can introduce light to that region. We can use light to control the neural activity. You guys feel okay with that? We just borrow the biology from algae. We can engineer in the mice, allow only this neuron that express this protein that normally not expressed in any brain, in mouse brain, any area. But we can use by engineering to express that. Then we can use light to control that. So that way we can use light to control the neural activity. So easy for us to see the behavior, right? So we can use different protein. We can specific to inhibit these neurons or to excite these neurons. In this case, I want to use light to inhibit this neuron. So we inhibit this neuron by light. And here I have this video. Let me see. So this mice, you can see the video. They their PVH neuron express this protein, and that can be inhibited by light. You can see I have the cable connect to the brain. That cable to deliver the light is to deliver the light to the neurons. Okay, so these mice. This early morning for mice is well fed. They are not hungry at all. So if I click it, so you can see the cable, the light is on, okay? Light on means the neuron is going to be inhibited. So the mice will engage in feeding behavior right away. Remember, these mice are not hungry at all. They just eat food during night. And as long as your light is on, the neuron is inhibited, the mice will always engage in feeding behavior. The longest time is a half an hour. They eat like 1 gram food. For mice, that's a lot of food. And use light is very powerful because once we stop light and we stop inhibiting the neuron, the behavior is gone. So it's on-off behavior, on-off. It's very, very a strong correlation, suggesting that inhibiting the neuron promote feeding behavior, no matter what. So it's okay, so far it's okay. I want to make sure, by the way, you guys, if you have a question, please ask, because I want to make sure that the information can be understood because of this, this is scientific, right? You have any question, you can ask. Okay, so just now I mentioned, for those obese patients, they eat food, they feel delicious. We want to also know whether this mouse, when they eat food, do they feel happy or not, right? That's all the information I know. So do this food, like eating, enjoyable to them, right? So for mice, it's very difficult. They cannot stand up and tell you I'm happy, right? We can design a assay. We can, using that assay, the mice will tell us through that assay. One of the assays called, we can do the same thing. We can stimulate inhibit those neurons using light. The way we did this, we put mice in this free arena, and we can pair 1/2 of the arena with light stimulation, right? Once the mice go to the on-side, the light will be on, so the neuron will be inhibited. Once the mice go to off-side, then the light will be off. There's no light inhibition. This is the control mice. The mice will stay equal time on-side, on off-side, because that's natural behavior. They must move around equally on both sides. But if you really to inhibit this neuron, you can see the mice spend most of the time in on-side, right? They spend most of the time in on-side, suggesting that they like it. Otherwise, they choose to stay that on-side. I mean, they like the neuron to be inhibited. They feel happy about it. That's why they spend most of the time in the on-side. So no problem with that, right? So they cannot tell us. They only can tell us through their behaviors. This is the behavior that tell us they like it, because they spend most of the time. They have free choice, right? They have free time on this side, that side, but they voluntarily to spend more time on the on-side, suggesting they like it. They like to be stimulated. So that means when they eat the food, they enjoy it. They feel happy about it. Just like our obese patients, they eat food, they really like it. So this is like similar to we human eating delicious food, right? Yes.
May I this light, it is simply a light that they're seeing visually. It's nothing that's attached to the brain.
This is we intentionally use a transparent so we can see light. So we can see when the light is on, when the light is off. But light can be delivered, actually is delivered to inside the brain. We perform surgery, we put a cable into the brain tissue and deliver the light to the neuron.
Oh.
We intentionally leave a transparent part outside so that we know when the light is on, when the light is off.
So
right?
this is stimulating the hypothalamus.
This particular is to inhibit that PVH neurons, a very tiny bit neurons, small number of neurons. That inhibit that neurons can be sufficient to produce all these like very compulsive behavior because they eat, they just eat. They don't care about anything else, they just eat. So then we want to see whether the other conditions that we talk about in our anorexia patient, they refuse to eat, right? So we want to see whether these can reverse that. Right? Those patients, they decide not to eat. They give up, they say, I cannot eat because that's caused too much trouble to me, too much of danger to me. So this is just this situation. So they have food, they need food, but they feel danger, they don't want to eat it. So in mice, we'll try to create a condition. Again, we use the same inhibition model, same paradigm, same thing. So we use a paradigm, we stress the animal. With this particular mouse, we spray a lot of water on their fur. The mice don't like their fur to be wet. They will groom extensively to get rid of the water from their body. So then we want, and in this condition, the mice will not eat, no matter how hungry they are. They are worried about their water on their fur. We try to use this condition to mimic the anorexia. Maybe not exactly match, but the behavior-wise, they are similar. So I can play this video. Let's see. Okay. So with the fur, they do extensively self-grooming, right? They want to get rid of the water, get rid of the moisture from their body. Look at the light is on. The light is on. See, light is on. When light is on, the mice automatically switch from the other side to this side to get food to eat. And this suggests that these are in a similar anorexia condition, but you inhibit this neuron, they're sufficient to reverse that condition. The mice now can start eating behavior. And very interestingly, so let's say, as long as the light is on, the mice will engage in. Light is off, the behavior is gone, the mice go back to the original location and start grooming again. So this is like a reversal process. So as long as you inhibit that neuron, the mice will engage in feeding no matter what, even in a situation that's similar to anorexia. And after that, they go back, resume the same behavior. So it's a very powerful way to make us eat more. Okay, for this anorexia patient, this can be I don't know how to achieve in human inhibit this neuron, but this can be a powerful way to do it. So the typical example that the mouse research can provide a lot of insights on our human biology, the principle. So then I want to see the reverse of that action. How about activation of this neuron? Just now I talked about inhibition, right? The activation of this neuron. So we can use same approach. We can use this way, but now we can activate neurons instead of in the opposite way. And here, I test this animal, this particular mouse, we fast them overnight. So this mouse is very hungry. They didn't eat food for a day for them. So let's get the video running. So they are very hungry. Remember, because they are hungry, they eat food right away because they feel like they're hungry. Okay. Now pay attention when the light is on. Light is on. Okay. Now light on. They stop eating right away. Okay. They as long as light on, They cannot engage in feeding. The light is still on. I want you to see when the light is off. Yeah, he's watching the food, but just do not want to eat. You see that? Remember, this guy is very hungry because they didn't eat for a day. The light is off. They remember they're hungry. They engage in feeding. So remember that. They just like, so we did this experiment for 20 times, on, off. Always follow that. Every time the light is on, the mice will stop feeding, light off, mouse engage in feeding. So this neural activation is again powerful, produce the opposite effect to inhibition, to stop feeding. So is this again this pleasant or unpleasant behavior for this mice, for this neuron to be activated? We did the same thing. We paired the light stimulation on one side and off on the other side. You can see the mouse spend most of the time on the off side. They don't like light to be on. They don't like this neuron to be activated. So they spend most of the time on the off side. You see that's clear. So that they don't like it. I mean, suggesting they don't like it. They don't like this neuron to be inhibited. And they don't like eating behavior because they don't like this whole feeding. It's a same thing, similar, not exact same, but a similar suggestion to anorexia. So as I said, you activate this neuron, they don't eat. So what they actually do, right? I mean, we're talking about in this anorexia, or they have this always associated with this mental disorders. So we want to also see whether they don't eat. What's their mental status? So in this way, we use same way to stimulate these neurons, but we didn't provide food. There's no food for them. These mice also not hungry. They're not starved. They're just normal. So we'll see what's their behavior. So now they're normal behavior because they are well fed, just normal. When light is on, you can see with behavior they engage. Extensive self-grooming. See that? As long as the light is on, they just keep grooming themselves. So I will talk about self-grooming a little bit. Self-grooming in animal is a very interesting behavior. The light is off, the self-grooming behavior is stopped, right? So this stimulation, you can see in 2 minutes period, some mice grooming like 60 times. It's a lot of grooming behavior. They cannot control it. So what is the self-grooming behavior in animal? So this is yes, exactly. So this is a coordinate that the y-axis is grooming pattern rigidity, the x-axis is grooming activity level. You can see there's a combination of two can be associated with a lot of mental disease. You see autism, you have a rigid patterning, you have higher grooming behavior. So all people, according to human behavior, they can categorize this self-grooming behavior corresponding to the human disease. And this grooming is also repetitive behavior. And repetitive behavior is a typical symptom of autism in human. Autism, you have communication problem, you have social interaction problem, and the other one is repetitive behavior. So when they engage in some behavior, they're always doing the same without any purpose. And these self-grooming is also the same. They are fine, but it's just self-grooming without any purpose. It's just doing it. And also, like some human, we have this called Autism Spectrum. Some of us like washing hands a lot of time, not necessarily, they're still washing hands, right? So this is similar to the human behavior that representing this autism spectrum. So self-grooming. So we also quantify the self-grooming in our animal, and we confirm that all these parameters change, suggesting the self-grooming through activation of this neuron also is anxiety related, is representing a part of autism spectrum compulsive spectrum. So we also explored how come, because there's a degree of stimulation here with a high strength stimulation. The light, we can use very high light, we can change their strength, right? The frequency, the power, we can change that. And here we also use a very high power of light to maximally activate those neurons. And you can appreciate it's very bizarre behavior we never saw from an animal. So now without light is on, very strong stimulation. So the mice, they never do that in regular basis. You can see, so the light is off, they stop that. I can play one more time, so you can appreciate how high the animal can jump. So this is 10 centimeters, right? We have a scale there. You can see how high they can jump. Oh yeah, of course, this must feel horrible, okay? But unfortunately, we need them to give us information about the brain. So this is, we have a lot of other assay that assay tell us they must actually feel fear. They are very fearful. They try to flee. This behavior is a fleeing activity. They try to jump out of the cage to get away because they don't like that. So this is the suggestion that activating of this neuron. So this is jumping behavior. Is that within 2 minutes they jump 40 times. Some of the animal after 2 minutes they just collapse. So they use all their energy during that 2 minutes period. So this is very strong behavior that can be controlled by this tiny bit of neurons in the brain region. Again, this is a fear and stress associated. Okay, so activation of this neuron can cause two distinct behaviors. One, self-grooming, you stimulate a little bit less, higher stimulation causes jumping, and it's actually a fleeing behavior. Okay, so now I just try to give a summary. So we'll talk about PVH neuron. You remember that PVH neuron. So we use this mouse model. The reason we use model, we can use bioengineering to monitor neural activity. That's the whole purpose. And if the neural activity is inappropriately inhibited, and the mice will engage in overeating, and is inappropriately activated, mice will engage in anorexia, like behavior. And this may have potential human relevance. I can tell you that, especially for these neurons. The reason we choose this neuron, we have reasons. So for that, I have to tell you another receptor called melanocortin receptor 4. So if you read a lot about obesity, human obesity phenotype, you will know this gene. This gene is most frequent monogenic cause for human obesity. What that means, it means that if you sequencing all the human genome and you separate the human population into obese, into non-obese, and this gene in obese will have the mutation. The most obese people will have this gene mutation. So this gene is so important for current human obesity. It's most frequent monogenetic cause for obesity. And if this gene function is disrupted, you will have obesity development. And this is shown here in animals. Sorry again. Yeah, for some, because we are all have different genetic makeup, and some of us have this mutation. Unfortunately, they will have a trend to develop obesity. And in a mouse, we can knock out this gene and see the mouse. You see the top curve, all knockout animal. They have double size than the controls. So this is gene function conserved in mouse and in human, clearly. So this is so important, and this gene is expressed mostly in these neurons, particular neurons. So suggesting, because this gene can activate neurons, now you have knock out this gene and the neuron will be inhibited. So exactly is a physiological phenotype for the behavior I just talked about. And I also have asked you to know another gene called, you don't need to know the name, but just this SEPAB3. And people made mice knock out this gene, and these mice show extensive cell grooming. You know, they grooming so much their nose also have a lesion there. Do you see that? That's from grooming. They just keep grooming. They know that these mice have this compulsive symptom like autism behaviors. And amazingly, and people cross, I mean, mice we can cross them, right? Where crosses two knockout together. And I can see this is amazing. It's amazing. So, on the top one is obesity phenotype. You can see the MC4R obesity gene, the knockout is very obese, right? On the left. And on the middle is Y type and SEPAB, this gene knockout. You don't see anything. But double knockout rescue the obesity completely. See, double knockout, the body weight is exactly the same as controls. So double knockout rescue this obesity phenotype completely. And another amazing thing is that double knockout also rescue this self-grooming completely. You see that the second mouse, SEPAB3 knockout mice, they have lesion, this extensive cell grooming, and double knockout normal. So double knockouts rescue both defects in those animals, and these genes expressed in PVH neurons. So do you see that? What's the chance to have double knockout completely rescue both defects 100%? So suggesting that they act on these neurons to have this function, to have this impact. I just want to use these two slides to tell you we have this potential human relevance. And because this is MC4, this is the obesity gene also occur in our human population. So now we try to understand whether this neuron is indeed underlying this behavior. Okay, just I think it is one of the final slides, just to sum up, give you a brief summary. So the PVH involved in this eating in both sides of the equation, and also regulate this emotion, and that can be underlying a lot of our either obesity phenotype or this anorexia associated with mental disorders. Finally, again, I want to thank, I didn't do this, my lab people did this, so all my lab personnel listed here, and support for various funding agency. And importantly, I would like to thank Cullen Foundation for their donation. So I'm the holder of Cullen chair. So every year I get a big chunk of support from them to try risky projects. Some of them was very risky. And if I propose, nobody can fund me. So I can use this money to do that kind of research. And then finally, thank you for your time, and also thank people online for their time. And I really appreciate this opportunity to share our research with you guys. And I will appreciate your question, because I feel sometimes we focus on animal research, sometimes feel like maybe some ways they're detached from reality, but your impulse can give me a lot of insights on the direction I should go. Okay, thank you so much.